Hi, everybody, and welcome to the first in a series of seminars from the Center for Leadership in Environmental Awareness and Research, the CLEAR Center. I'm Melissa Runge-Morris. I'm the director of the Institute of Environmental Health Sciences at Wayne State and of the Environmental Health Sciences Core Center called CURES. I'm here with my colleague, Dr. Carol Miller, professor of Civil and Environmental Engineering. Let's go to the next slide, Brendan. So we're going to hear a few introductory slides today about the CLEAR Center because it's all new. And also most of our time today will be spent with the 5 individual project leaders that form the heart and soul of the CLEAR Center. CLEAR is the first Superfund Research Program to be funded by NIEHS at Wayne State University. It's a very large transdisciplinary center grant, as you can see, with a total value of $11 million. And I have the pleasure of co-leading this program with my colleague, Carol Miller. Next slide, Brendan. All right, why do we need a Superfund Research Program at Wayne State University? Well, Wayne State University has a very strong urban mission and a very strong connection with understanding and solving the problem of environmental health disparities. And as reported annually by the March of Dimes, it's noted that Detroit has the highest preterm birth rate among 100 comparable American cities. So that's a real emergency. In Detroit, we deal with urban legacy and emerging contaminants, and these contribute to a variety of health consequences, including adverse reproductive health effects. Now, the focus of CLEAR is on volatile organic compounds, or VOCs. These are a common contaminant in Detroit's subsurface and Superfund sites from industries that we've had in our city. And the CLEAR team postulates that VOCs are an important determinant of adverse birth outcomes. We believe that Wayne State University is uniquely qualified to lead this investigative effort. Next slide, please. Go ahead, Carol.
Yeah, thank you very much, Melissa, for kicking us off. And I want to welcome everybody here to the seminar today. As Melissa mentioned, I am from the Civil and Environmental Engineering Department. I also lead Wayne State University's Healthy Urban Waters program. And we think this is an excellent pairing of engineering, environmental science, and the medical and health fields to deal with this problem of subsurface VOCs in urban areas. The map you're looking at is showing a variety of features that are very important to our Superfund Center. And although I won't go into detail, I do want to make the point that much of the work, especially on the engineering and environmental sciences side, that will contribute to the CLEAR Center will have some foundation in geospatial mapping, that is, looking at relationships between various variables throughout the city. And this map is showing locations of varying degrees of preterm birth rates. And it's also showing sites of known subsurface VOC contamination. Certainly our engineering projects, as well as our biologic and epidemiological projects, will be gleaning lots of information from relationships that you can see. Next slide, please. So the primary mechanism of exposure to VOCs that we're looking at within the CLEAR Center is attributed to vapor intrusion through foundations of homes, apartments, or even businesses. As you can see in this cartoonish slide here, we're looking at subsurface contamination in the groundwater. That's considered a saturated zone of the subsurface. The contaminant will evaporate or volatilize out of the groundwater and will then have an upward trajectory due to energy gradients that lead it towards any sort of cracks in the basement of a home foundation or any sort of building foundation. So this is the mechanism for the contaminant getting into the home. And another thing that is being highlighted in this particular drawing is some of the complications that come into play, especially in urban areas where we have many sorts of anthropogenic materials that are in the subsurface. Not natural pristine soil necessarily, but things like sewer lines, electrical cables, piles for foundations for buildings and other structures, as well as, of course, legacy contamination. Next slide, please. This is a graphic of the organizational structure of our CLEAR Center at Wayne State University. And what we're depicting in this slide is the integration between the 5 projects, which are here labeled Engineering 1, Engineering 2, and Biomedical 1, 2, and 3. Really, we have 5, as you can see, 5 research projects. And those span all 5 of our cores. So on the right-hand side, you can see the 5 cores that are listed that come together to solve the research questions within each of the 5 projects. Next slide, please. Now, what you're going to be learning is a bit more about each of the 5 projects, 2 of those being engineering and environmental science, and 3 of those being biomedical projects. We're going to kick it off with Project 1, presented by Dr. Glenn Hood. Next slide, please.
Good afternoon, everyone. Thanks for the introduction, Carol. Like she said, I'm Dr. Glenn Hood from the Biological Sciences Department. I'm the lead on Project 1. I'd like to introduce our other team members here quickly. We have John Lucas Baroni. He's co-PI on this project from the Department of Environmental Science and Geology. We have my PhD student and CLEAR trainee, Sarah Black. She's from the Department of Biological Sciences as well. And our team is expanding. We just hired 3 new undergrads, and we're going to be bringing on an additional grad student and a postdoc soon. Next slide. So Melissa did a really good job introducing the problem here. Detroit has the largest preterm birth rate among major US cities, and this has been linked to exposure to volatile organic compounds. And so the goal of Project 1 is to build above ground strategies to identify and address below ground hotspots for VOC vapor intrusion in complex urban environments. And our project is divided into 3 aims. Aim 1 is to develop methods to use above ground plant tissues to identify VOC hotspots. This is a methodology known as phytoscreening. Our aim 2 is to use stable isotopes of water molecules to identify below ground source water for urban plants to isolate possible sources of VOC contamination. And then our third aim is to verify below ground presence of VOCs and link in that we want to link to indoor VOCs. Kind of for the sake of brevity, I'm going to focus on aims 1 and aims 3 today. Next slide, Brendan. So some of you may be thinking, why would we want to use plants to screen for VOCs? We have traditional methods. You can sample water from a well, for example. Well, there's a high cost associated with drilling and maintaining wells, but perhaps more importantly, there's limited access to private properties because drilling wells to sample water is highly invasive. So you can imagine that maybe a private property owner, a private homeowner, or a business wouldn't want someone installing a permanent or even temporary well on their property. So in comes this idea of phytoscreening, and it's a rather simple idea. It's to use chemical analysis of plant tissue to provide evidence for below ground contamination. And the way this works is really simple. Plants grow this below ground network of roots. And those roots are absorbing water that they transport to the above ground portion of the plant, the stems and leaves that plants use for photosynthesis. And VOCs, depending on the particular type of VOC, they are partially or completely dissolved in water. And when they're locked up in water, they're less volatile. So it would kind of stand to reason that at contaminated sites, if there's VOCs in the water, the plants are going to be uptaking that contaminated water as well, and potentially transporting it to the above ground portion of the plant. And so I would argue that phytoscreening for VOCs, specifically VOCs, is really in its infancy, but we do have a growing number of examples that are effectively using phytoscreening. Next slide, Brendan. So here are two such examples. One is from Gopalakrishnan, publication where they were using willow trees to detect concentrations of TCE. This was published in 2007, and our own publication published a couple of years ago, where we were using grapevines to detect a semi-volatile organic compound, 1,4-dioxane. I think there are two take-home messages from this slide. The first is that, sure, you can use plant tissues to effectively detect VOCs, but maybe more importantly, it looks like there's quite a bit of variation depending on the plant tissue that you use. And that really speaks to what we're going to try to do in the first couple of years of Project One as it relates to phytoscreening. Next slide, Brendan. So we're going to partner with Luogen, the Chemical Instruments Corps here at Wayne State, to develop more nuanced, detailed, and thorough methods to actually extract VOCs locked up in water that are in the plants that will allow us to more effectively screen plants for VOCs. The way we're going to do this is we're going to go out to targeted locations here in Detroit where we know they have an exceedingly high concentration of VOCs. And we're going to sample plants, different species of plants, different plant tissues across the growing season. And we really want to try to answer what I consider a fundamental group of questions that will allow us to further develop this methodology. And I've just listed a small subset of these questions here, but you can imagine why they're important. So, for example, the different plant species that are common in Detroit, do they have different phytoscreening potential? Some of our preliminary results show that grapevine would be a really great candidate, but there are other plant species at the same highly contaminated sites that could that detect VOCs in much lower concentrations. Another important question is, do different plant tissues within a species have different phytoscreening potentials? And so the data on our previous slide suggests that may very well be the case. Another fundamental question, what time of the growing season is optimal to sample? And perhaps maybe even more important than that is, how are climatic events affecting VOC concentrations? Some of our preliminary results suggest that after a hard rain, VOC concentrations decline. Perhaps that's because the rain is diluting the below ground VOCs, or they could just be being washed out completely from our sites. Then, of course, we want to address fundamental questions about the depth of the root system, the type or structure of the soil. But once we hone and perfect this methodology by answering these really important questions, next slide, Brendan. Our goal is to go out and phytoscreen Detroit. And we're going to partner with the Community Engagement Corps, the CEC, as is led by Mike Thompson, to do this. And so what you see in this map are homes, potential homes that Mike and his group are going to go out and sample the indoor air for to detect VOC concentrations. And so what we want to do is sample the plants in the yards of these homes to see if we can link below ground concentrations to the concentrations of VOCs in the air inside these homes. And so on the right here, we have some preliminary data that speaks to the power of this approach. We collected this with Andrea Cassidy-Bellows and Jennifer Strawn. They're from the Henry Ford's Public Health Science Department. They're leading Project 5. You'll hear from them later today. But what we did is we went out and phytoscreened or sampled the plants in the front yards of 13 homes of pregnant women throughout Detroit, and we screened them for VOCs. And what we have here are the results from those 13 homes for a particularly common VOC, toluene, but it still has negative health outcomes. And at each home, we sampled 6 different plant tissues from different plant species. And the thing that I think is striking about these results is that within a site, there's quite a bit of variation, but there's also quite a bit of variation between sites. So hopefully with the methodologies and our experimental approaches that we're going to be doing in the first couple of years of CLEAR, we will be able to answer this question. Why is there such a large amount of variation? So I thank you for your time this afternoon. And I think next up is project 2.
Hey, good afternoon, everyone, and thanks to Melissa, Carol, and Glenn to introduce overall center and project E1. And I'm Yuni Uyghur, and I'm associate professor in the Department of Civil and Environment Engineer at Wayne State University, and I'm leading the Clear Engineer Project 2 together with the other project co-lead, Dr. Tim Dietrich. So the overall goal of our project focus on the real-time monitoring and remediation of vapor intrusion. So currently, it's very challenging to timely test and remediate vapor intrusion, particularly in Michigan, where a large population live in close proximity to many vapor intrusion sources. For example, as shown in the sad news, you can see that for nearly 20 years, a family has been in a risk of being exposed to toxic vapors without knowing it. Next, please, Brandon. So why detecting and remediation vapor intrusion is challenging and lagging? One reason is that vapor intrusion can be affected by many factors, such as geospatial variations, seasonal and weather changes, and house conditions. As a result, like shown in this table in the left side, the level of exposure risk to the vapor intrusion can vary dramatically from tens to thousands times of difference. So for the conventional methods of testing vapor intrusion is very labor intensive, time consuming, and expensive. It requires trained professionals to go to the site, collect multiple air and soil samples, take it back to the lab, and analyze the different VOC concentrations. So the whole testing process can take weeks to months with a high cost of a few $1,000s for just one site. And the remediation process is even more expensive and time-consuming, with many engineered practice and operation being involved. So these challenges could cause many sites remaining untested or missing harmful exposure window if not tested at the right time, as shown in a figure in the right corner in a figure, show in those red sampling dots. If we go to collect those samples at the time that the concentration is below the threshold, and then we will miss the detection of the harmful exposure to vapor intrusion. And also it's hard to assess the effectiveness of the remediation, how effective the remediation due to the limited test. Next slide, please. So this project, it is a problem-based and solution-oriented research by integrated the innovative research and practical solutions to address the challenge of testing and remediating vapor intrusion sites. So the first research aim and the solution is to develop a novel real-time vapor intrusion monitor system by integrating the Internet of Sensor and edge computing. So compared to conventional offline testing and other sensor monitor approaches, the advantage of our monitor system that including the low cost, that our system can also continues to do a real-time data collection and monitor that can minimize the missing of detection of harmful exposure windows. And it's also easy to install and use. And then we plan to, we have very good promising preliminary results. We validated those sensors and to assemble those different sensors and hardware, and also implement some algorithm together to collect the data at real time. And we also, right now, we also validate it in the lab. So right now we improve those sensor system. We plan that in the 3rd and 4th year of the project, we will apply our sensor monitor system in over 100 Detroit homes. So we will work with a community engaged core to go out to testing in those 100 homes. The second research aim and the solution is to develop the vapor intrusion prediction model and app by using the data collected in our research aim 1 from over 100 Detroit homes, as well as data from other CLEAR project and center and open data sources. So this prediction model and app will be used to predict vapor intrusion exposure risk in various locations and house conditions. It will serve as a screening tool to identify potential vapor intrusion exposure hotspot to prioritize vapor intrusion tests and remediation in those high-risk areas. So the third research aim and solution is to develop a novel solvent-based remediation system. We mostly will focus on air filter, use the novel solvent that we developed. Those novel solvent has advantages or benefits with high sorption capacity and no desorption, so that it can avoid the second contamination from the filter. And we will also integrate the remediation air filter with our real-time vapor intrusion monitoring system to trigger the operation of the air filter whenever it's needed. And at the same time, we can use real-time monitor data to evaluate the effectiveness of the remediations. Next slide, please. So far, we have made some very good progress for our project with very promising results that have been published in this peer-reviewed journal papers. So due to the time limit, I will not go too much in detail, but if you are interested to know more about our project progress, please contact us or go to visit our newCenter website. Next, so at the end, I would like to thank our wonderful project team that including multiple investigators and trainees from Wayne State University, North Carolina State University, and University of Delaware. And I would also like to thank our students who have already graduated, but during their time studying with the university, they have made a great contribution to this project, like such as those preliminary research, those preliminary results. So thanks for them. Again, I thanks everyone to be here in the meeting for the opportunity to share our project. As the next, I would invite the project 3 leader to share their project. Thanks.
Great. Thank you, everyone. Nice to see everyone. So I'm Tracy Baker. I'm the project leader for Project 3. Excited to talk about our project on behalf of our interdisciplinary research team. So along with myself, Dr. David Pitts, Sean McElmary, and Thomas Backhouse, encompassing environmental toxicology, environmental engineering, pharmaceutical sciences, and ecotox. We also have some great trainees that are already working on the project, Mackenzie, Zoha, Emily, and Dima, ranging from undergrads to PhD students. So our project is using the zebrafish model to look more in depth at the mechanisms and biomarkers that these VOCs are causing. Our aims are first to evaluate adverse health outcomes. So we're looking at developmental, immune, reproductive, and neuro outcomes from both acute and chronic exposure to these various VOCs at different levels. And then a big thing that we want to do is look at the effects of different mixtures of these chemicals that I have listed here on the right. And we're able to do that in the zebrafish model, and then look more specifically at gene regulation as well as potential epigenetic changes that might be happening due to these different contaminants. So, we use the zebrafish model, as I mentioned, mostly because, we're interested in trying to do a more high throughput model. They can develop very quickly. And so by 5 days, all of their structures are formed and functioning. They can also reproduce within 3 months. The females can lay up to 100 eggs per week. And also the genome is sequenced. So that allows us to do some of this analysis as well. Next slide, please, Brendan. So what we want to do is one of the unique things about VOCs that they do volatilize, and what makes them difficult to study in some systems. So we use specific vials, and then we've also created this specific chamber in order to keep the VOCs in contact with the fish. And we're looking at a number of different compounds based on concentrations that are already reported in Detroit. And then we'll also be getting concentrations from the projects you already heard about in Project 1 and Project 2, as well as humans in the community to look at, again, a number of different concentrations, which is the beauty of the zebrafish model. Next slide. So we've already started some of this work, and we have some of it published where we've been looking at benzene at a few different concentrations. We're going to go back and look at more of those. We didn't see, at least at the concentrations we looked at, we didn't see mortality or, changes in different phenotypes, a few different changes in abnormalities. But what we did do is we were able to identify some key genes and pathways that are involved in reproduction, as well as, a number of different genes that are involved in cytokine response, which you'll hear about in a little bit, in inflammation, that actually overlap with some of the placenta samples and some preliminary data from the mouse exposures. Next slide. So we raised these fish up. So what I just showed you were the larval exposures. So we're also looking at these long-term exposures. So we're looking at the adults. And so again, these same benzene exposures, we were able to look at the brain and gonad tissue. And again, we saw a lot of overlap in different pathways that were involved in endocrine system as well as reproductive system, and a lot of overlapping pathways as well. On the next slide, we've also started looking at 1,4-dioxane, again, kind of using this same method, and have seen, some other different phenotypic changes. And then in the next slide, again, you can see that, in addition to cancer, which we know 1,4-dioxane can cause, we're also seeing these endocrine system disorders and a number of different genes that are involved, that we know are also involved in human reproductive pathways. So next slide is just wanting to acknowledge the many people that helped with these different projects, and I'll pass it off to Project 4.
We're actually going to transition to Project 5, and then Gil will move to your project after that.
As you wish.
Thank you. Brendan, can you move to the next slide? Thank you. So I'm Jennifer Strawn. I'm going to present on Project 5 on behalf of myself and the MPI in this project, Dr. Andrea Cassidy-Bushrow. We're both based at Henry Ford Health and have been really diving deep into some of the epidemiologic studies around preterm birth. And our project then focuses on volatile organic compound exposure during pregnancy and preterm birth. Next slide, please. So up to this point, I think we've heard a lot about VOCs, and I just wanted to add a little bit of context and tell you more about preterm birth. Preterm birth is really defined as birth at less than 37 weeks completed gestation. It's associated with both immediate and long-term negative health outcomes for infants, including things such as an increased risk of infant mortality and an increased risk of cardiovascular disease, among other things. And so it is obviously a very important health problem to understand and even try and prevent. And while you've heard some of my colleagues mention that Detroit has the unfortunate distinction of having the highest preterm rate among large cities in the US, they didn't really yet mention the fact that there's another concerning statistic, that there's this persistent racial disparities in preterm births, and the preterm birth rate among Black women is 54% higher. Oh, Brendan, I'm sorry, I think we jumped way ahead. There we go. Sorry. So that's okay. So that I was saying the preterm birth rate among Black women is 54% higher than among non-Black women in Michigan. And in Detroit, we have a really diverse population and a very high preterm rate, a high percentage of women who identify as Black or African American. And so despite all these statistics and despite a lot of research, few strategies have really been successful at reducing preterm rates. And so I think that's where we're kind of investigating other things that might play a role, such as volatile organic compounds. Next slide, please. So some of the early work by our study team has found that VOCs are associated with preterm birth, specifically in Detroit. This study in particular used geospatial data and estimates of BTEX exposure, and BTEX is one type of volatile organic compound. And really found that for every 5-unit increase in BTEX, there was an increased odds of preterm birth, and that odds ratio was about 1.5. However, this study did have some limitations that our study in this Project 5, I guess, will address as we move forward, and that we estimated BTEX exposure. So we did not have direct estimates of volatile organic compound exposures, which could lead to some exposure misclassification or bias. Next slide, please. We also looked at a couple of mechanisms by which BTEX might influence risk of preterm birth. One of these was looking at inflammation. So we know that a successful pregnancy really requires tight control of maternal-fetal interaction in the immune system, and any disruption of that, which could lead to preterm birth. In this study, we found that TNF-alpha and IL-1 beta, which are both pro-inflammatory cytokines, which were dysregulated, and that could lead to an increased risk of preterm birth. This study was also done in Detroit in an exclusively African American population, but the sample size was smaller, and we weren't able to really look at some of the other details that we'd like to look at, such as preterm birth subtypes, or again, even individual level measures of VOC exposure. Next slide, please. One of the other pathways that we've been really interested in understanding is the role of the placenta. We've started some pilot studies looking at placental DNA whether that placental DNA methylation is altered in response to BTEX exposure, and even trying to look a little bit at changes in gene expression in the placenta. Our early studies do suggest that there might be something going on, and that we need to look a little bit further into that, even with the small sample size that this study had. Next slide, please. So this slide really presents a broad overview of our proposed project. In Project 5, we'll be directly measuring VOC exposure in maternal biospecimens across pregnancy. We're hoping to capture early, mid, and late pregnancy measures. We'll be specifically interrogating 2 different pathways, looking at maternal inflammation in response to VOC exposure, as well as looking at placental DNA methylation and transcriptomics, and whether either of these pathways, might lead to that increased risk of preterm birth. We're going to be collaborating with the various cores that are involved in this project, and I think it's really exciting that we'll be able to look at some of the pathways in different models. So looking at what we see in terms of gene expression and inflammatory markers, both in zebrafish and human studies, and as you'll hear next, in mouse studies. That is all I have for here, and I'll hand it over to Project 4. Thank you.
Okay, perfect. Thank you, Jenny. My name is Gil Moore. I am the PI of Project 4 in collaboration with an incredible team of experts in the field of toxicology and environmental that includes Doug Gruden, Mike Petrioli, Mariana Sadagorski, and Jiahao Ding. The next slide, please. The first concept that I would like to introduce everybody is that whatever happens in the environment during pregnancy is going to have an implication in the health of the mother as well as in the health of the baby. But I don't want to make all the pregnant women worry that they're going to do something wrong to the baby, because in reality, nature has developed mechanisms that the response to the environment, the first thing that they do is to educate the baby. And they educate the baby to something that is called resilience. So if the mother gets infected with the virus, it doesn't mean the baby is going to get sick. Or if the mother is exposed to environmental things, it doesn't mean that the baby is going to be abnormal. I think it's the opposite. The mother, in the majority of the cases, is going to be sending signals that we call maternal immune activation, for example, and it's going to educate the baby into the good thing, that when the baby is born, it will be able to deal with the environment. The next slide. Unfortunately, when the stress of the environment cross this threshold of good education, then things start getting bad. And one of the aspects, and thank you to my colleagues in the previous presentation, so I don't need to go that, environmental factors, especially when there is a major concentration of abnormal levels of environmental factors, those lead to diseases and multiple types of diseases. It has been defined air pollution as the silent killer. And there is the area or the field of the impact of air pollution in children's health, who has also been focused mainly after the baby is born. But now we have a strong data, and there is a growing data, that what we see in the baby really is the outcome of what was happening during the time of the pregnancy. The next slide, please. So what do I mean? And again, this is the work done by this fantastic group. I don't need to repeat them because already you heard from them. Let's pass all this. Also, the next slide that already was presented by Jennifer. Next slide. So all what is leading this is that environmental factors are going to induce maternal immune activation or maternal inflammation, if you would call it. And all they're supposed to do to the resilience, if they cross, as I mentioned, this threshold, they are going to induce abnormalities in the development of the fetus, who then will be translated in multiple types of diseases, depending also on the target. And that can be early stages of the health of the individual, for example, asthma, abnormal responses to vaccination, abnormal responses to infection, and even in later age of the baby or the offspring leading to cancer. So the focus of our study is to understand these non-infectious stimuli, that is the environmental factors during pregnancy and the implications into the mother and the offspring. Next slide. So the question is, what is here of our grant, what is the impact of maternal immune activation on the fetal immune system? Next slide. So the first question that we started, and by the way, I just want to mention that all this and more has been now published in iScience. So that article will be coming hopefully next week, and then I will share it with everybody. So the first question that we have was, does benzene exposure induce maternal immune activation and inflammation? That's what we wanted to know for the beginning. Next slide. In order to answer the question, we needed to develop a model where we can expose to environmental factors, to VOCs, especially benzene, and study. And of course, we cannot do in humans, so we went to mice. Next slide. The model was developed by Dr. Ding and the PhD student Anthony Maxwell is using this chamber system where we expose pregnant mice from the beginning of the pregnancy to the end to benzene in different concentrations. So the data that I'm showing you is mainly with 50 ppm of exposure to their pregnancy for 5 hours. Next slide. Up, you went too fast. What? One more back. One more back. Yeah, yes. Let's go back. Now I lost you. Yeah, here. No, one back. Come back. One more. This is the important one. This is the important. Okay. No,
okay.
One more. Yeah. No.
Sorry.
Slowly, don't kill me. Okay. So the first thing is we wanted to know, we exposed the mice to benzene. Is this either having at all an effect on the pregnancy? And interested enough, and in correlation with the findings that you hear from the human studies, the exposure of benzene is inducing preterm birth. Now, in the mice, those who are not mice doctors, I need to remind you, there is no preterm birth. It's called reabsorption, because the mice has multiple fetuses. So when the animals were exposed to benzene during the pregnancy, we see an increase of the reabsorptions, increase, of course, in the pregnancy losses. And although we started very early, we did not see that there was an impact on the number of implantations. In other words, did not affect the normal implantation and the normal pregnancies. It was mainly inducing an early termination of the pregnancy. Next slide. Now we will go less. And as was mentioned before, we found that this early termination of the pregnancy was associated with maternal immune activation or maternal immune inflammation. Inflammatory cytokines or high levels of inflammatory cytokines were detected in the blood of the dams, of the mothers. Next slide. So the message now, the first message that we have from our studies is that benzene induces maternal inflammation and has an impact on pregnancy outcomes. Next slide. So the next question, of course, was what is the target? If there is a maternal inflammation, we are seeing the termination of the pregnancy. What is the target? Next slide. And of course, the first site that is connecting the mother with the fetus is the placenta. So we look, is the maternal inflammation also triggering an inflammation in the placenta? Next slide. And indeed, that's the case. When we take placentas of mice exposed to benzene, those placentas are expressing high levels of pro-inflammatory cytokines and very, very strong inflammatory cytokines, such as IL-12 and interferon gamma. Two of them, and again, we have additional data, including interferon TNF-alpha and IL-12, what are the triggers of the process of parturition. Next slide. Okay. So how the placenta responds to these dangerous signals, especially to benzene. Next slide. That's the data that I want to show you that it has been published now. So the placenta really changed when we expose the mother to benzene. And there is significant activation of genes in those placentas, both increase or decrease. This is RNA-seq. Now, I'm going to jump already and tell you that if you put all the placentas together, you really don't get a clear picture of what is happening. But if we separate male and female placentas, then it's amazing or it's so intriguing the differential response that we see in a male placenta and a female placenta towards the same stimuli that has been seen. Next slide. So again, this is the showing you that there is major changes, but the interesting aspect is the changes are not the same in a male placenta compared to a female placenta in response to benzene. Next slide. To summarize this, again, the first message that we're finding here is that although they are exposed to the same stimuli and in the same mother, a male placenta and a female placenta are responding totally different to benzene, to the pollution. Something very important to take in consideration when we want to look at the outcome of the offsprings. And again, because of time, I'm not showing that data, but we have very interesting results showing that males are more susceptible to abnormal responses when they are exposed to benzene. And I think I want to show you the next slide. I will pass these slides very quickly. Again, benzene and controlling the male, I just mentioned major changes. The aspect that I am putting this slide is to remind me that the changes in the male placenta in response to benzene are associated with immunological genes, while the next slide, the changes in the female placenta are associated with metabolic changes. So if we want to understand what is happening in the offspring later, we have to take in consideration that the male has different changes than the female. Next slide. And with that, I finish. Let's pass this because I just mentioned. Let's go to the last slide. This is the grant that we are studying. So we have been able to show that benzene-induced maternal immune inflammation, that inflammation is reaching the placenta, is inducing inflammation in the placenta, but is different between males and females. And now the objective of this grant is to look what is the outcome in terms of the newborns to either environmental challenges or bacterial and viral changes. I just can tell you that we are finding that males from benzene-exposed dams are more susceptible to viral infections, and that's not the case to female offsprings. Again, as I mentioned, the data is now published. I will be happy to share with you next week. And in the next presentation, I will be delighted to share with you also our findings now with the offsprings. Thank you.
Thanks, everybody. So this is just to summarize very broadly that through the CLEAR team, we are looking for how to tackle a very tough environmental health problem. We're using the approach of interdisciplinary research and training to identify the sources of very important environmental health disparities in our city. And you heard from the horse's mouth in terms of all the PIs of these projects, 2 very innovative engineering projects, and 3 symbiotic, really complementary biomedical projects to address the complexity and very urgent problem of the effects of the VOCs in our urban built environment and how these exposures affect preterm birth and other very important health outcomes. Shall we open the floor to questions?
Sure. Yeah, we have 13 minutes, it looks like, left within our scheduled time. So feel free to toss any questions into the chat. I haven't seen any myself yet. But if anybody wants to either raise their hand or just unmute.
I have a question. I guess it's for both Gil and Jennifer. Are there any sex differences in the human epidemiology data in terms of preterm birth for humans exposed to BTEX, like Gil is seeing with the mice?
I think with Jen, we have had that conversation. I'll let you answer that, but that is, I want to bring the point here that we need to find a mechanism that allow us the share of data between Henry Ford and Wayne State. We have it stock for almost 6 months just to try to find a way how to collaborate and to share information. Jen, go ahead, please.
Yeah, Gil, I think there's been significant progress made, so I'm really excited about that. But yeah, you know, Doug, there's not a lot of research out there on VOCs and preterm birth. And, you know, while we know that, you know, maybe males are more likely to be born preterm, and, you know, Gil's findings and some others suggest that, you know, placenta respond differently if they're from male or female babies. In terms of human studies, I don't think that's really been fully investigated. It's definitely something that we need to look into further, and hopefully we can do as part of this collaboration.
Yeah, it would not only be preterm birth, but child outcomes if there's a sex difference.
Correct. And I wanted to add that to what Jen said. If you look at the data, for example, of children in Detroit with asthma or with sensitivity to respiratory infections, is the dominant male compared in terms of female. So more boys have respiratory complications and asthma compared to females. And that's what we're seeing also in our model, that the male is a hypersensitivity to respiratory stimuli. The female, on the other hand, which is a complete different area, and we'd love to discuss this in more details with Jen, they have metabolic changes. And I don't know really what it means, those metabolic changes that we're seeing, how it's translated into the girls' metabolism. Obesity. We were talking with the pediatricians, and obesity in the children in Detroit, and in a study with asthma, the average of those children had a BMI of 90. It's amazing. It's amazing.
Looks like Melissa has her hand raised. You're muted.
Sorry, Glenn, could you take a few moments to talk a little bit about what we've discussed before in terms of how do you handle the fact that in screening plants themselves also generate VOCs as part of their biological activities? So how do you handle identifying the appropriate controls for the studies you're doing?
Yeah, right. Thanks, Melissa. That's a really great question. My honest answer is I'm still figuring that out. I met with Judy Restrick, who runs the Lumigen Chemical Core, to determine if we can actually do that. So for example, those results that we presented with toluene, there are a lot of plants. I don't think any of the ones we sampled, but there are a lot of plants that produce toluene naturally, and some other VOCs, and almost all plants produce some VOCs. They use that to attract pollinators, for example. Or they can use it to attract pollinators, for example. But from preliminary talks with Judy, we should be able to chemically decipher between those naturally produced VOCs from plants themselves versus those things that are pollutants that were in the groundwater that are being taken up by plants. And so, I think long story short, stay tuned, but we should be able to do that relatively easily. And I'm getting a handle on what those methods are and what it would require.
Yeah, thank you. That's interesting work that you're doing. Thanks.
Yeah, thank you.
It looks like we have a question from Cameron in the chat. Given that many of these compounds are found together, any movement towards studying combinations of common contaminants?
Isn't that what you're doing, Tracy?
Yeah, so using the zebrafish model, we're able to, again, because they're more high throughput and we have, larger numbers, we are going to be looking at several different mixtures, both what is in the environment that we already know, and then what all the other groups are finding, and working together with, we have a mixture specialist toxicologist that's part of our team.
And yes, also for us, the second phase will be to start now using combination. We are trying to focus first with benzene because we also want to understand mechanism of action. It's much easier to dissect the mechanism of action only with one compound, with one VOC than with the combinations.
Thank you. It looks like Carol has her hand raised.
Yeah, thank you. My question is for Glenn, Dr. Hood. My question relates to procedures that he and his team might be using to isolate the VOC impact on vegetation to VOCs that are coming from the subsurface, as opposed to VOCs that might be in the ambient air or high levels of air pollution on any particular day or any location.
Yeah, so that's in regards to AIM 2, which I didn't present anything on, kind of like I was saying for the sake of brevity, but our goal is to try to use stable isotopes to tease that apart, whether they're coming from groundwater, it's coming from lower underground, or from soil moisture in the subsurface, or whether they're coming from moisture and VOCs that are ambient in the air. And the stable isotope analysis of water molecules will allow us to do that. But it also might allow us to determine the source of where these VOCs are coming from. So not just air versus different subsections of the below ground, but where the actual source is. And so also in combination with Project 2, where they're going to be doing some groundwater modeling, we might also be able to figure out how they're moving underneath the ground.
Thank you.
If I may ask to this group, I don't know, from the epidemiologist, is there any information about miscarriages and environmental factors such as VOC in Detroit on the area? If somebody knows about that.
Yeah, hey Gil, this is Jennifer. I actually am not sure about anything with regards to miscarriage, especially within Detroit. I haven't really looked into that literature.
You're probably asking that, Gil, because of the, that's how health outcomes are signified in the mouse model you're working with, right? So maybe miscarriage rate would be another piece of the puzzle relating this to human health outcomes, right?
That's correct. So we have an interesting new data in vitro and some of the in vivo that the exposure of some of the metabolites may have an impact on trophoblast function. And if it's early, will prevent the invasion and migration of the trophoblast, who may lead to early termination of pregnancy. So that is the reason I must, again, this is preliminary data. We would love to know if somebody has information about a correlation with miscarriages. Again, preterm birth and miscarriages is almost the same, but just in different times in terms of the process. But although there is another mechanism, it's a simplification. But it is an interesting observation that we are developing. Thank you.
Yeah, Gil, just to follow up, I think it's really, this collaboration is really exciting because we have the opportunity to look at some of these pathways in different models and translating, what we see in human studies and what you see in mouse studies to human studies and kind of back and forth. And even letting our, like I know someone asked about combinations of contaminants and really taking what we see in humans and using those levels, applying them in mice or zebrafish, or what's seen in plants. I think it's really neat that we can work across the projects like this. I'm really excited about it.
I agree with you, Jennifer, because we can move from the overall analysis into more hypothesis-driven or pathway-driven, and then you start focusing on those specific genes. And we have some of them, so I hope soon we can start having regular meetings and we can show you that data.